Welcome to Unit 16, my pretties. Today we're going to talk about polymerase chain reaction, or PCR, and molecular techniques. This is a real short lecture, and I hope you do enjoy it. Here we go, Unit 16. So nucleic acids are essential to life, basically, and nucleic acids consist of many nucleotides in a long chain. And there are two types of nucleic acids, including DNA and RNA. DNA is the double helix molecule that carries genetic instructions for growth, development, and reproduction, while RNA acts as a messenger carrying instructions for DNA for controlling synthesis of proteins. Single-stranded, shorter chains of nucleotides would be how you would describe RNA. Humans have 46 chromosomes arranged in 23 pairs, and of course one pair are the sex chromosomes. And genes are sequences of DNA carried on a chromosome that encode information for the translation of nucleic acid sequences into amino acid sequences that result in the production of proteins. Whew! Nucleotides form the basic structure for DNA and RNA. They're linked polymers of pyrimidines and purines, which are also known as base pairs. They're called a base pair, especially if you have something like adenine and thymine that are connected by a hydrogen bond. That's called a base pair. A polymer is a molecular structure made of similar type bonds. But again, we have the nucleotides that are purines and pyrimidines. We have nucleotides made of purines or pyrimidines, or both. Purines are adenine and guanine, and pyrimidines are cysteine and thymine in DNA, and cytosine and uracil in RNA. Nucleotides are bonded together, and two strands are twisted into a helix, forming DNA. So replication of DNA involves one strand of the molecule acting as a template for the creation of the other strand. Hydrogen bonds hold the strands together, forming the helix, but these bonds can be broken, and that's called denaturation. Denaturation, and we're going to talk about that in PCR. They can also spontaneously rejoin, and that's called annealing, and we'll talk about that in PCR as well. Functional proteins are made from DNA through transcription and translation. We have RNA, which is easily replicated, and there are three forms: messenger RNA, transfer RNA, and ribosomal RNA. Okay? Messenger RNA conveys genetic information to the ribosome, while transfer RNA mediates recognition of the codon and provides a corresponding amino acid. A codon is basically three base pairs in a row. Ribosomal RNA makes proteins from the ribosome. One of the greatest advances ever in medicine is PCR, or polymerase chain reaction. It can actually amplify a single copy of DNA into billions of like copies in 25 to 40 cycles. The principle of polymerase chain reaction is that we take minute amounts of specific DNA and amplify it so it can be further analyzed. A target sequence of DNA must be known in order to use this technology. This sequence needs to be between 100 and 1,000 base pairs in size. Okay? A short portion of that target section of DNA is used as the primer. An even shorter section of DNA, around 16 to 20 base pairs, is going to be our primer. These become the template for making our new DNA copies. In order to analyze products of PCR, we use gel electrophoresis or real-time PCR using fluorescent dyes. All right, we'll try this again and see if this link works for the PCR procedure. If not, I'll go ahead and post it. It may not, since I'm narrating these PowerPoints, but we'll see. So copying DNA is an enzymatic process, and that is carried out in three different cycles. Those cycles are called the denaturation cycle, the primer annealing cycle, and the primer extension cycle. Okay? In the DNA denaturation cycle, basically strands of DNA are unwound, and this occurs in about 15 to 30 seconds at 95 degrees Celsius. The primer annealing cycle takes place at about 45 to 65 degrees Celsius, and that takes about 30 seconds. And then finally, primer extension is to make more products by adding the enzyme Taq polymerase at 72 degrees Celsius. New nucleotides are then added. Okay? Having gone through these cycles, specific DNA doubles exponentially. 30 cycles of amplification is typically the mean number of cycles for adequate DNA amplification in order for us to study that little bit of DNA a little bit more specifically. There's enough genetic material at this point to analyze through another method like gel electrophoresis, probe hybridization, direct DNA sequencing, or dot blot and reverse dot blot, and we'll talk about those here momentarily. Remember, amplification of PCR in microbiology is used to diagnose bacterial, viral, fungal, or parasitic reactions or infections, or to determine bacterial resistance. So that's how we use PCR in microbiology. In a conventional method, we use gel electrophoresis where bands are separated based on charge. Nucleic acid bands are then dyed and fluoresced using UV irradiation. Okay? Then we have DNA sequencing, which is an enzymatic process where complementary DNA to that being sequenced is used to identify the DNA indirectly. Probe hybridization is combining a known labeled nucleic acid probe of genetic material with that derived from the patient or unknown sample. This will also include the formation of hydrogen bonds between nucleotides of RNA and DNA. In liquid hybridization, we have the target nucleic acid and labeled probe interact in a solution. In dot blot and reverse dot blot, we are going to use that to identify diseases by the analysis of a mutated sequence of DNA. Okay, so there's three different types of blot type of methodology. We've got Western blot, Southern blot, and Northern blot. In Western blot, we're using it to detect mutations and identify microorganisms. In Western blot, proteins are actually separated electrophoretically, transferred to a membrane, and then identified through the use of a labeled antibody. In Southern blot, DNA is denatured and the fragments are separated by electrophoresis, and the fragments are added to a membrane and radiolabeled. And then in Northern blot, we have messenger RNA that is separated electrophoretically, and we can actually detect RNA in the transcription phase here. So these are the methods used to analyze amplified DNA from PCR.